the cost of that one training example?
Ah, you haven't been able to hear me for 5 minutes. That's so annoying. Let me just check this now. Hello, hello, hello. Hello, hello, hello. Hello, hello, hello. Oh, that's junk. Okay, so for the last 5 minutes I've been talking to myself. That's good because it didn't make a lot of sense. So, first of all, I was away a bit. First of all, I was away a little bit longer because I was watching the video from 3 Blue 1 Brown, who's excellent at explaining things. And so there was a couple of mistakes I made. The mistake was that this L is not an exponent, it is an indices. And the reason he's put it into superscript is because subscript will come later and he requires both indices. Cool, okay. So that's number one, my apologies. Number two is that um, I'll just go through this a bit, although I do recommend you just watch his video. He says that if you take a normal neural network, you would have a couple of, you'd have your input, and then you've got a couple of parameters, and then it comes out as the output. The output is the cost, right? C, and the cost is by the cost is the sum of the weight, the bias, the weight and the bias, and the weight is this dot here, and the bias is this link here. Okay, and this is 1, and this is bias 1, and then you've got the bias 2 and weight 2, and so on and so forth. And so the cost is the output of those. So this whole function takes in the back propagation of all the weights and biases accumulated. Okay, so what he's doing here is he's just putting it into one dimension, so it's much easier formula. And then he goes into the second set of indices, the subscript indices. So we'll get to that. So here he's saying that A, which is the weight of layer 1, and then you've got this kind of let me just actually draw it here. So you've got this uh, the weight is here, and the weight is this output, whatever. And then you've got this connection here, which is the bias, and then you've got the second weight, and this is layer -1. And the reason it's going backwards is because that is the back propagation, I think. So this comes out as 4/8, whatever. And then the desired output is denoted as y, where y is a cost. So y is the desired output of 1, which is that the derivative equals 1 because there is no angle between them. And then, I don't know if you can see this. I think you can see this, maybe. And then you've got this whole thing becomes the weighting minus the last cost squared. That's what that means. And that is equal to the new cost. So this is C0, and you run this function over and over again, and that looks like that. Cool. And therefore, if you summarize like exactly what he's doing here, the ZL equals, so Z becomes the cost is put into a function for L. So, or is summarized by Z of layer 1. And the cost is obviously then the weight of layer 1 multiplied by the weight of the last layer. Whoops, didn't mean to do an extra A there. That's what that means. And then it's multiplied by, oh no, sorry, plus the bias of layer 1. So that's So L is this one here. L minus 1 is the back propagation, and then the weight. So why is it weight? Where does the W come from now? Where is the W coming from? Let me just see what it says.
WL times
Right.
As a reminder, this last activation is determined by a weight, which I'm going to call WL.
Okay. Mm. So the weight is in the middle, I see.
Times the previous neuron's activation plus some bias, which I'll call BL.
Okay, that makes sense, doesn't it? So actually, this is the weight.
Training example as C0, a weight, which I'm going to call WL, times the previous neuron's activation.
Activation, that's why it's called A. So I can't remember what the activation refers to now. Okay, that's interesting. So, okay, fine. So we'll call these activations on layer -1. Plus this bias. Bias, I guess. Damn, you have to watch the video. It's cool, but I'm terrible at explaining it, but I need to understand this myself. So if you've got this whole function gets compressed into this, where you've got the weight of layer times the activations. What is the activation? Let's find this out again.
Is y. For example, y might be 0 or 1. So the cost of this simple network for a single training example is AL minus y squared.
AL minus y squared, so 0.66 minus y, the output. The desired output, I see.
We'll denote the cost of that one training example as C0, 1, decrease to the cost function. And we're just going to focus on the connection between the last two neurons. Let's label the activation of that last neuron with a superscript L, indicating which layer it's in. So the activation of the previous neuron is A.
Activation. So there's this kind of concept of activation, and now I'm a little bit confused at what that's referring to now. Let's go along with the maths here. So anyway, so this whole cost function is called now, or labeled Z for that layer. Z Z for that layer. Okay. So here it is. And then you've got the next layer. So he's now calling this AL. Is equal to the application of that sigmoid function, for example, the sigmoid being that non-linear function, or whatever you want to do, okay, times the last cost. Or as a, no, so you pass into that the last cost, I see, right. And then if you go down in graph form, let's see this. So if you go down in graph form, you've got the cost output of layer 1, I guess, is the result of the last output times the activation of L, and then you've got the last cost output, which has inputs of the weight, the activations of the last layer, okay, L minus 1, yep, and then the bias, that link in between the where B is like that. Okay, that makes sense. Right. So here he's denoting that just like calculus, you're now trying to find out that if you have all these biases, the weight of L, yep. Well, so bias, the activation, and the weight, you multiplying them together here, because this is the cost. Because you're multiplying them together, they're very sensitive, and so the cost is like massively altered depending on that sensitivity. So he's saying this is the derivative. So the derivative he's put on a graph here for us, and he's saying if you change this number, then that obviously sways quite a lot, and together they sway a heavy amount because they're all multiplied by each other. So this is the derivative, and that's why then I think he goes on to express it as a differentiation equation. This, dy over delta cost 0 over delta weight of that layer. That's differentiation there. And that differentiation, oh, this is interesting, yeah, because he's saying it's equal to the ratio. Yeah, okay, so he's chaining every single, I say, let me just do this. This delta of the last cost function divided by the change in the weight of that layer. So it's the last layer divided by the new layer multiplied by the change in the activation of this current layer divided by the change in the last functions or last cost multiplied by the change in this current cost divided by the change in this layer's activation. So it's, let me just say it again because that's quite amazing. So you've got the, okay, let me do this here. This is output, let me just do this here, like that. Output of last cost divided by this layer's weight multiplied by the derivative of this activation, this layer's activation, divided by, or a ratio of, the output of the last cost, the change in the last cost, and then you've got the change of this layer's cost as a ratio of this layer's activation is equal to the change in this. Ah, look, no, this is 0 and this is sub-0. Let's listen to this. So
derivative of ZL with respect to WL. Likewise, you then consider the ratio of the change to AL to the tiny change in ZL that caused it, as well as the ratio between the final nudge to C and this intermediate nudge to AL.
The final nudge to C as a ratio of well, I'm not sure what this is denoting actually, in all fairness, but I can see that the cost of this, or the change of this layer's cost over the change in the weighting of L is, so the change in this layer's cost divided by the change in this weighting of this layer divided by the change of weighting in this layer. So the cost is proportional to the changing of the weight in this layer. Oh my god, right, yes, okay, I see. Does this make sense to anyone else? Because that just made sense to me. Thank goodness. Okay, wow. Equals the change in the output of the last cost function, which is proportional. No, it's a ratio of this layer's weight. So the last cost is divided by, because it's a ratio from the last output of the cost function. No, I see. Okay, so this, oh my god, the last cost is a ratio of the weighting of this layer, because the weighting of this layer is going to skew that last cost, and then it's proportional because it's multiplied. Proportional, is that right? I think that's the right word. By the activation layer, the change in the activation layer, divided by the change in the last cost of the last layer. So you're multiplying that together, and then you've got the change in the current cost divided by the change in this activation layer. Alright, let me just run through this one more time, because this is important. It's trying to build a picture of how things, yeah, so this is called the chain rule, how things affect each other over that period of steps that we do. So if we're going through, everything is affecting the next one. So this is why Z super L is the output of the cost. That's got to be tied into the new costs. So the new cost, or the change in that new cost over the change in this layer's weight, and this layer's cost in fact, is equal to the last cost output, all proportional to this whole thing, this whole layer. I'm explaining this terribly, aren't I? Next episode, sorry, is really good, because I think this will settle in my head. Beautiful, anyway. Okay, so he gets to this point, and he's saying that this is why the cost output is so sensitive to all of it, because it is all in proportion to each other and in ratio to the weighting outputs of the costs and the activation layers themselves. So they're all heavily integrated into this equation here, which makes sense. Now this one goes on to say that so he's saying that the change in the cost of this last change of cost divided by the activation of that current layer is equal to 2 times the activation layer minus y. Yikes, yikes. Let's try this. So let me just write it myself. Sometimes I write it and it kind of makes some sense to me. So the change in, fuck this thing, I hate that bloody symbol, delta, change in 0 over the change in alpha of L, alpha L, which is this indices, right? So this current layer, the change in this current layer, and the little tweak in the last cost is equal to 2 times the activations. Oh, because there's 2 activation layers. Oh, I see. Okay, that makes sense. 2 activation layers minus the output or the cost. Minus the cost. Okay, fine, that makes some sense. And then so the cost of C sub naught equals 2 times, so it's going to be if you're squaring, so the derivative, oh man, feeling cogs working at the moment. I'm feeling the cogs work. 2 times this. This is some algebraic shit going on here. Okay, well we'll just trust that. I need to learn my algebra. I think that's what's happening there. That's why I'm so confused. And he's saying that
L with respect to ZL is just the derivative of our sigmoid function, or whatever nonlinearity you choose to use.
Yeah, okay.
And the derivative of ZL with respect to WL, in this case, comes out just to be AL minus 1.
In this case becomes AL minus 1. So the change in the current cost over the change in the activation of this layer. By the way, layer is equal to this. So this is layer 0, 1, 2, for example. I think you can add more layers as you go along. So he's talking about this first layer minus the output of the actual minus the cost, y being the cost output here. Okay. And so this cost is equal. Oh my god, yeah. Because y represents the last output, whereas cost is the output of this whole thing. The cost of this is the new cost to apply to the next layering system of backpropagation. Yes, okay. Oh my god, that makes so much sense to me now. Right, yes. So if I say it, maybe this will make some sense. So y is the cost of this set of equations, and so y, or at least the cost, is equal to this set of equations, which equals that cost, which would be the last cost. So this new cost would be the sum of that. No, maybe that's not right. God. Okay, let's just go on. So you're getting all these derivatives, great, okay. Weighting of this layer. Yep. Okay. Oh yeah, and this new indices things, K and J, I think he's got going on, is to denote when you have an extra indices to work with, basically. So if you've got let me just read all this again. If you've got your original dimensional neural network and these added another indices, that means that the function now becomes, instead of just activation of L, it's activation of L over J and K sort of thing. So okay, that makes sense why he's got that. Now he's saying derivative of full cost function is equal to 1 over the current number from 0 to the sum of all of that from k equals 0 to n minus 1. So we're going to go backwards through the loop. 1 minus n. Okay, 1 over n. Wait, can I just draw that? Let's get some Desmos here. Desmos! Oh, that's a sign up, I didn't know that. Desmos Graph. So if I go x equals 0, let's do minus 5 to 5, step of 1. And if I do 1 over x. Oh, ha. X equals, oh no, what have I done? X equals 1 over x. Oh. X. Just making it 1 divided by 4 is going to be smaller and smaller. So x is going to be... Okay, maybe I just do yeah, n, sorry. And it's 1 divided by n. So as it gets bigger, it gets smaller, great. And as it gets negative, it gets smaller too. Cool. So it's really reversing. So it's kind of getting closer as the number skews away. Okay, that's interesting. Oh man, if oh damn, what's his name? Is it Chris? No, it's not Chris. Who does 3 blue 1 brown again? I can't remember. It is Grant. Yeah. If Grant watches this, he'll be like, "Lol, who is this guy?" So it's saying that this will approach 0. Is it 1 divided by n, because it's going to get smaller as as 0, and it's going backwards through that loop, and then it's multiplied, no, as a function of the change in c over k equals 0 minus 1, and a change in the weighting of this layer. Average of all training examples. Okay. It's hard, isn't it? Okay, so this is some gradient matrices, I think he says, because it's going to go constant bias, weight of L, BL, weight of 1, B1. Wait, so this is, so okay, so the change in the cost is equal to a matrix of each layer. Is this layer? I wonder what the 1 represents, so maybe I should watch this again. Let's just watch this part.
Is just one component of the gradient vector, which itself
Gradient vector
is built up from the partial derivatives of the cost function with respect to all those weights and biases.
Let's listen to that again.
And of course, that is just one component of the gradient vector, which itself is built up from the partial derivatives of the cost function with respect
Partial derivatives of the cost function. Okay, that makes sense.
to all those weights and biases.
I don't understand this expression 1 and then the L. I guess where L is whatever layer and 1 is the first layer. I don't know.
But even though that's just one of the many partial derivatives we need, it's more than 50% of the work. The sensitivity to the bias, for example, is almost identical. We just need to change out this del z del w term for a del z del b.
Okay, oh my god, okay. As it iterates through, which route does it come down? Oh man, that's amazing. Okay, cool. So on and so forth. And I think he adds this, this J indices for moving in X directions, or J direction, or K direction, whatever. And then, here it is, which route that it took. I guess it takes every route, because it has to go through each one. Yeah, it does. Okay. What's this? Recap. Yeah, okay. Let's see what this... So the change in the cost is n layer plus 1 minus 1. N sub L. So the number of the layer plus 1. So we're going through each layer. And then going backwards. What? I don't understand this at all.
That determine each component in the gradient that helps minimize the cost of the network by repeatedly stepping downhill.
Stepping downhill. So stepping backwards is... It's got to be backwards. This negative 1, right? The layer plus 1. J is 0. Okay. The sum of all the weighting on each indices or each segment of that layer. The layer is plus 1. So we're going through the layers forward. You're doing the sigmoid to the sigmoid being that function to the output of the last cost, L plus 1, the cost of which line of outputs multiplied by the change in this cost as a ratio of the change in the activation of whatever route it took. Oh, I'm not sure. Or 2 times activation. What was J again? Let's just try and get J. This is one of those things, literally the next time I watch it, I'm going to be like, oh my god, this makes so much sense now. J and K. Okay, so J is...
to Z. You can kind of see what I mean, right? Where all of these are essentially the same equations that we had before in the 1 neuron per layer case. It's just that it looks a little more complicated. And indeed, the chain rule derivative expression describing how sensitive the cost is to a specific weight.
It's just because it's interesting because J and K has been joined up here, but J and K, so J is this part. Weight is oi oi oi. Oh my goodness. Right. Okay, I need to watch this again. That was fascinating. So coming back to this, let's not forget this, and optimize these parameters using gradient descent. Gradient descent is literally this diagram here, because we are trying to descend down this gradient, this matrices, this vector, this gradient vector, to try and get to the lowest dip in that graph, which would be the most precise match without overfitting. Gosh, wow, it's fun, but it's way over my head with maths. Okay, cool. So we've got this code. Yep, we've got this all done. I don't know if we've got the forward part. Let me just check that. Not there. Let's go PyCharm. So in this case, we knew up our neural network, and our neural network uses this flatten, which I can't remember what it does. Let's see, flatten. Flattens a continuous range of variables into a tensor. A continuous range of variables into a tensor. Okay, so NN, flatten it, stack sequentially. 28 times 28, 512. Rectified linear. ReLU stands for. It's not that, is it? Rectified linear. Oh, it is. Rectified linear activation function. Okay. Short is a piecewise linear function that will output the input directly if it is positive, otherwise it will output 0. Okay, so it normalizes it. Piecewise linear function that will output the input directly if it is positive, otherwise it'll output 0. Yes, I remember that. Cool. And then this linear, and then writ linear. Okay. Right. Let's just go with the flow here. And then hyperparameters. Hyperparameters are adjustable parameters that let you control the model optimization process. Different hyperparameters values can impact model training and convergence rates. We define the following hyperparameters for training: number of epochs, the number of times to iterate over the dataset. So we were just about to draw this, I remember. Let's go back to this. We were going to have the number of so we define the following hyperparameters for training: number of epochs. So this is going to be for 0 to n, or we were calling it E for epochs. Number of times to iterate over the dataset, batch size, the number of data samples propagated through the network before the parameters are updated. So that is for each layer. So that's activation of L. Let's call it L of n, or ln, like that. And then, well, actually, let's just do this sub until we need it. Super. And then we've got number of data samples propagated through the network before the parameters are updated. And then learning rate, how much to update models parameters at each batch or epoch. So this is a function of this. Smaller values yield slower learning speed, while large values may result in unpredictable behavior during training. So how much you cut back. So this is how many layers you're going to go through, how many weights you're going to go through, how many activations, biases, yeah, how many activations and biases, that's what I want. So smaller yields, okay, cool. So the learning rate you can actually just set, cool. So that is E equals 5. It's not going to be the sum of as well. This is all proportional, so it's just like, is it integration? I can't quite remember. Maybe something like this, I don't know. Define integration, maths. Technique of finding a function gx, the derivative of which dgx is equal to a given function. An integral assigns numbers to functions in a way that describes displacement, area, volume, and other concepts that arise by combining infinitesimal data. The process of finding integrals is called integration. Integral assigns numbers to functions in a way that describes displacement. So it's not the integral, it's certainly not that. Function E. Is it the sum of all this? It's going to be the cost is equal to the cost of n. Well, anyway, this is basically stepping from n to or 0 to n, and we're going backwards, n minus 1, and then it's the output of this function. So let me go back to this, or let me just look this up. Mass iteration symbol function. F of function. Oh god, let me just do f. So the formal definition of iterated function of a set x follows: let x be a set and f x to x be a function. Defining f of n as the nth iteration. Defining f of n as the nth iteration of a function, a notation used by Hans Heinrich Berman and John Friedrich William Herschel, where n is a non-negative integer, by f def idx and f of n plus 1. Definition, what's this def? Where idx is the identity function on x and f og denotes function composition. That is, the function composition times x is equal to f of g of x. Always associative. Because the notion fn may refer to both iteration, composition of the function, f of exponential of the function, the latter is commonly used in trigonometry. Some mathematics choose to use to denote the compositional meaning, writing for the nth iterate of the function. Iteration sequences, limiting behavior, invariant measure, fractional. So how do I do differentiation, iteration? Trying to figure this out. Well, I'll do this another time, but you get the point. So this is running this over that iteration, and then let's go back to this. Yeah, okay, so let's put these bad boys in. Let's go here. Once we set our hyperparameters, we can then train and optimize our model with an optimization loop. Each iteration of the optimization loop is called an epoch. Each epoch consists of two main parts: the training loop, iterate over the training dataset and try to converge to optimal parameters. The validation or test loop, iterate over the test dataset to check if model performance is improving. The test loop, the training loop and the test loop. Let's briefly familiarize ourselves with some of the concepts used in the training loop. Jump ahead to see the full implementation of the optimization loop. The loss function. When presented with some training data, our untrained network is likely not to give the correct answer. Loss function measures the degree of dissimilarity of obtained result to the target value. And it is the loss function that we want to minimize during training. To calculate the loss, we make a prediction using the inputs of our given data sample and compare it against the true data label value. So here's the thing about this. We need to, so I'm just trying to think of the bigger picture here. I need to know, let's imagine I wanted to, measure the time between, measure the time it takes for a nerve to complete one circuit of execution. Now, I guess that is from point of input to brain output. This is a terrible diagram. Let me try this again. Input of this sensor, some other n sensors, and then the brain output or brain received. So if I wanted to know that, I need to get data on this. So I would say human h of n, and this is 0 to nerve. Let's call that RV. Let's call that V, nerve. So 0 to nerve. A V is how many nerves it would go through, or nerve connection points, or each. So if an input goes up through your nervous system to your brain, are there any junctions? Let's call them junctions. So I don't know this, but I don't think that there is like one specific continuous point. It would amass through a section, or it would amass through a collection of sensory points, because you don't, like if you hurt your hand, you can also feel it up a certain period of. Okay, well we'll just figure that out, but let's call that junctions. So then it would be human of n. So we'd have to have a collection of humans of this function, data, and then we'd want to, so let's imagine we're trying to measure how long it takes for the nervous system to execute one input to the brain, right? So we need a collection of data of that input, that specific input, across a multitude of humans, and then we'll get a set of results. So n equals 0 to n equals 0 to n. Yeah, okay. So because we need to train the algorithm to say, well, so then what you'd need to do, so this, let's say this is for, okay, this is good. So let's imagine this is for, let's go, human in like assumed optimal state. Like, I'll say that because I know that we're all very, but at least the function of that human is like, I feel fine. And then you would take someone with I don't know, let's say it's got diabetes, or I've got diabetes, so that's good. Human with diabetes, and you'd measure. Let's call this all output or output. And so we'd want to get what's the change in let me just think about this graph. So we've got this graph here, and this would be h of n, or this would be human, or okay, time taken. So let's call this. So the output of this function is t for time taken. Cool. And so t is that way, and then you've got what's the change in that based on state. So state is this whole, so S is this whole state thing. S equals the state. So state. So if you've got, I don't know, it takes humans it's kind of like the time it takes depending on the state. So it would be, if the state was, it would have to be different graph. This isn't going to work. I want to have the state is going to be here, because you'd have like, I don't know, diabetes, and you'd have Alzheimer's, and you'd have broken arm, and then how much time it takes would be that as an average of all of them, and it would take that long, and then it would take that long. So now you've got this graph coming where it'd be something like this. So this would be the time t. Whoops. Let's go for a different color here. But this is not variable because it's the time taken. But this is the change from different. This isn't the. Is this the change? This is the difference between, yeah, different states of the body. So we'd need data for. So data required for. Data for D, A, and B, etc. And then we'd say we take someone's measurement of their neural productivity, and then we would fit it. But that would be too precise, because like if you get the output, let's say that there is that's a difference between, you know, like from here to here and from here to here. There's like a range. this would be beautiful to find, right? If the range overlaps, so if this one comes down here, then how would we know that it's not this one? Well, it'd have to be based on other states. So then you would have to say, what are the other states at play here? So maybe you could take blood as well. Then we're getting some squiffy correlations. But let's say we take this bigger now. Let's say we take a bit more. So for state n equals state of body, or no, disease state, I guess. Whoops. And then you've got blood state, blood measurement. Now this is good because blood has a lot of things you can measure. And then you've got breathing. Let's call this gassy. Breathing measurement. You know, these are all abstract thoughts at the moment, obviously. And then we'd have biological markers, let's say that, things like that. Biological markers. Now, let's say that there is Z of these. So now we'd want so S becomes an ever-changing thing, but it's not changing per stat. So it's going to be D A B all the way to Z. But this is no longer T, it's variable. Let's call this V for variable. And so we can use a gauge theory for this. If we go, yeah, okay, so we'll do gauge theory for this. So that can be changeable variable. Now, in gauge, so I need to, gauge theory, is that the one? Like a fiber bundle kind of thing. Okay, what I need to do then, I think the next thing, in terms of actually thinking of the bigger picture, how do I correlate V in this? And I'm pretty sure there's a notation in maths for that, which will be really helpful because then I can start to structure formal notation around what I want to achieve. I need to gather a clearer picture of what I want to do as well. I need to know where I can source kind of some test data, or can I come up with some test data? Yeah. Yes, that's what I need. I just put some test data in for now and see if I can get some algorithms going, and maybe I can do it with chemical data. So yeah, here's the problem. So yeah, T, it would be time of disease state, or the time of neurological. Oh no, but would it change? Yeah, okay, so it would be it won't change because t is still going to be the time which that nerve takes to get to the brain, but it would be just recorded over a series of different states. So what's the blood measurement, blah blah blah, you'd say B, blah blah blah. You'd have this massive graph of data to work from. So we don't need gauge theory here. Yeah, that's good. Okay. And then what's the time? I need some food. Okay. So when we've got that, we can start to figure out some AI around that. We can use test data. Now the data's going to be I wonder as well, because we could take things like scans. I don't think I'll be able to get access to scans of a human body essentially in its entirety. That's not going to be possible. I might be able to get access to some brain reading stuff. Maybe, if I'm very polite and kind. I think with the accumulation of all this data, I might be able to start picking away at the problem of finding something specific that denotes an ailment. Now, here's the thing. I wanted to use CRISPR too, which is to modify function to create essentially a 3D model. So by 3D model, I mean if I wanted to take a copy of the nervous system and then allow the body to create that copy next to it somehow, I would need to then analyze. Okay, so here's task 2. This is like T1. And this is wrong. This is wrong. We don't need V. I'm just going to cross that out. V remains T. Cool. So let me take a picture of that. Move on to the next one. Yep. Next one is, if I want to take CRISPR, I need to know. Let me just think here, because I think let's go to Twitter. Grab up some of Manolis Kellis's work so far. Manolis Kellis. So this is free information he's providing here of his team's work. Yeah. Let's go open this in Firefox. Right. So you have things. Oh, bit low quality that. You might not be able to see this. This has got a little bit difficult to read, to be honest. Evolutionary stuff for primates, carnivores. Okay. Experimental fat mass. So all these differences. So it's got, yeah, at the bottom. B is comparison of energy deprivation score between allometrically scaled fat mass and experimentally measured fat mass across mammals. C, organs with high fat content, organdroxide, whatever you say, human specific module variation, metabolic processes, transcriptional regulation, MAP kinase, lipid degradation, white adipose tissue, human specific module variation, transcriptional regulation, mitochondrial organization, catabolic process and lipid degradation. Organs with high fat equals this, and this is adipose tissue equals that. So you've got diagrams here. Network derived discovery of biological processes 28 in human enriched gene sets from tissues with high fat content. Data derived from differential expression by RNA sequence and ATAC sequence comparing human and primate tissue. Okay, so I think we've got these kind of things we can delve through, which is like essentially papers scientific papers. And in terms of CRISPR, what can we learn from that? What can we do with that? If I were to take a nervous system, you know, as lovely as it is, and we take a copy of the DNA that creates that, so we'll call this nerve and then nerve 2. Now this discovery of what genes create the nervous system, genes that create nervous system. Now we then want to do we want to know where attachments are. So we've got, let's call this attachment, or okay, so output for brain, input N, N1, N2, I, N to Z, or just dot dot dot. So we need to know all the inputs. Okay. Genes that create genes that create the nervous system. We take all the inputs. So we need to understand every single input of the nervous system to then be able to, well, that's interesting, because do we? So here's the question. Do we need to know all of the inputs of the nervous system? Or are they emergent based on other parameters? Because if you take the creation of a baby, okay, so that is DNA at the start will denote its output, although there is expression. So we need to know what part does expression have? What part does expression have to play on the above? Because if this is all denoted in the sequence at the start, how do we then study how do we study the DNA of adults without the input of expression? Do we need to take baby DNA? Question, not sure. To remove expression noise. This is, I mean, I don't know this stuff, so I'm just thinking out loud here. So if that's not the case, let's say we can take the expressed version at age X, age Y, then what are we then thinking? So let's say, so we've got like I know the DNA of all 30-year-olds, let's say. Then you want to find the part that denotes the nervous system. Now this might already be studied, but if it's not, then how do you know? So how would you know what protein function creates a nervous system? You would have to use AI. Yes, Okay, wow. Okay, so you'd use AI to run through different protein function outputs and see which one expresses the nervous system. Holy shit. Wait. This is good. Okay. You might have to use AI to run simulations of the protein functions to see the output of their see their output. If an output looks like a nervous system, bingo. Okay, so here is the AI coming in. Alrighty, we finally got there. That took not very long. So AI, boom. So here's the question: what do we already know from AlphaFold's? Let's look at AlphaFold's data. Do they present it to us? AlphaFold data. Let's see what they've got. DeepMind. Okay, got it. In our new, when was this updated? 15th of Jan. It's quite old. Okay, 2020. In our study published in Nature, we demonstrate how artificial intelligence research can drive and accelerate new scientific discoveries. We built a dedicated interdisciplinary team in hopes of using AI to push basic research forward, bringing together experts from the fields of structural biology, physics, and machine learning to apply cutting-edge techniques to predict the 3D structure of a protein based solely on its genetic sequence. So, this is interesting because they mention biology because of the functions of cells, etc. The physics because at a quantum level what happens if you add something, then it would have different electrons, it would have an effect. This is kind of chemistry. And machine learning is the application to formalize and process data to find patterns that denote stuff. Now, okay, so yeah, because then you don't have to do the experiments up front, which are probably expensive and take time. To apply cutting-edge techniques to predict the 3D structure of a protein based solely on its genetic sequence. So here it's not just the protein function, it's actually the sequence that produces the protein function. This allows us to inject function in. Our system AlphaFold, described in peer-reviewed papers now published in Nature and Proteins, is the culmination of several years of work and builds on decades of prior research using large genomic data sets to predict protein structure. The 3D models of proteins that AlphaFold generates are far more accurate than any that have come before. How accurate? How accurate are AlphaFold protein outputs? Median score of 92.4 GDT. Informally considered to be competitive with results obtained. What does that mean though? GDT. Global Distance Test. The main metric used by CASP to measure the accuracy of predictions. CASP is that competition, isn't it? Our competition of what does CASP stand for? Something like that. CASP. Critical Appraisal Skill Program. Okay. Predictions is a global distance test which ranges from 0 to 100. In simple terms, GDT can be approximately thought of as the percentage of amino acid residues, beads in the protein chain, within a threshold distance from the correct position. Oh, so they're saying that they are very accurate. Where is it saying it again? Oh, in that website I saw. Okay. AlphaFold 2, score of 14 is 90 something percent. Holy shit. Okay, wow, that's mesmerizing. Right. Okay, so yeah, we were trying to look for data that they've got. This paper, our system, described in peer-reviewed papers now published in Nature and Proteins, is the culmination of several years of work and builds on decades of prior research using large genomic data sets to... Okay. The AlphaFold code used at CAS13 is available on GitHub here. For anyone interested in learning more or replicating our results. Okay. Let's have a look through this. AlphaFold 2, please. Guess I haven't released that yet. CAS13. What was this one? CAS14? I think it was. Where's the damn it. Oh, here it is. CAS14 2020. This is 18. Okay. Aye. Is there CASP? Maybe there is one. CASP? No. Ah, that's so irritating. That is so irritating. Okay, fine, cool. Thank you very much, guys. Any publication provides an implementation of the contact prediction network used in AlphaFold 1. Associated model weights in CASP13 dataset as used for CASP13 2018 and published in Nature. It's a completely different code from that used in AlphaFold 2, which was used in CASP14 2020. Any publication that discloses... Okay, let's just ask first. I need to go... Well, I'll ask later. If I can have AlphaFold 2. I'm guessing that might be, like, not allowed yet, maybe not ready for it to be released. Paper actually can be found on Nature. Okay. Improved protein structure prediction using potentials from deep learning. Using potentials from deep learning. Protein structure prediction can be used to determine the three-dimensional shape of a protein from its amino acid sequence. This problem is of fundamental importance as the structure of a protein largely determines its function. However, protein structures can be difficult to determine experimentally. Yes, they take like years, or a year. Considerable progress has recently been made by leveraging genetic information. It is possible to infer which amino acid residues are in contact by analyzing covariation in homologous sequences. It is possible to infer which amino acid residues are in contact by analyzing covariation in homologous sequences. It is possible to infer which amino acid residues are in contact, by analyzing covariation in homologous sequences, which aids in the prediction of protein structures. Here we show that we can train a neural network to make accurate predictions of the distances between pairs of residues, which convey... someone's figured this out. Look, oh god, this is brilliant. To make prediction of proteins, here we show distances between pairs of residues. Someone had to find that out. That is a correlation, wow, which convey more information about the structure than contact predictions. So the residue left behind is that, which convey more information about the structure than contact predictions. Okay, using this information we construct a potential of mean force that can accurately describe the shape of a protein. This is the physics part. that can accurately describe the shape of a protein. We find that the resulting potential can be optimized by a simple gradient descent algorithm to generate structures without complex sampling procedures. The resulting system, named AlphaFold, achieves high accuracy even for sequences with fewer homologous sequences. Damn, guys, I am so sorry. I am so sorry. I just want to absolutely be sure I know this word now. Having the same relation, relative position or structure. Cool. With fewer similarity sequences. With fewer similarities. In the recent critical assessment of protein structure prediction, CASP 13, a blind assessment of the state of the field, AlphaFold created high accuracy structures with template modeling scores of 0.7 or higher for 24 out of 43 modeling domains, whereas the next best method, which used sampling and contact information, achieved such accuracy for only 14 out of 43 domains. AlphaFold represents a considerable advance in protein structure prediction. We expect this increased accuracy to enable insights into the function and malfunction of proteins, especially in cases for which no structures for homologous proteins have been determined. Right, where's the paper? Paper, please. Do I have to buy it? Where's the paper? Access options, rent or buy, get limited or full article access and recube. Our training, validation and test data splits are available from the following versions of public datasets we used in this study. Oops. CASP 14. Have you found it? Is this it? No, it's not. What does this say then? Let me just read that again. Our training, validation, data availability. Our training, validation and test data splits, domain codes, are available from GitHub. The following versions of public datasets were used in this study. This is important. Right, okay. Shit, I'm out of honey. More honey, 6:43. Do I dare to continue? How long have I been doing this? Yeah, an hour 30. I'm getting somewhere though. Do I stop now? One more honey. I'll be right back. Cool, cheers. Okay, right. Let's have a look through that code then. How long? 3 months ago. This is quite old stuff. Okay. This package provides an implementation of the contact prediction network using AlphaFold 1, associated model weights and CASP 13 datasets as used for CASP 13 2018 and published in Nature. It's a completely different code from that used in AlphaFold 2. Okay. The paper abstract can be found on Nature's site. Let's just have a look at that. That's the one we just had a look at, right? Yeah. Abstract. Cool. And the full text can be accessed directly here. Yes! Okay. Yes! Right. We got it, we got it, we got it. Great. Let's read this then. Uh, sure. Oh, how do I like this? How do I start this? Let's just start this. I'm not even signed in here. Let me just go and do this on Chrome. Sign in. Yep, that's me. Let me just start this so I don't lose it. Start. Right. Good. No, don't click that. And this is going to go in my little doody here. Yes. Alright. Improved protein structure prediction using potentials from deep learning. Legends. Shout out to these guys. Protein structure prediction can be used to determine the three-dimensional shape of a protein from its amino acid sequence. Yes. This problem of its amino acid sequence. This problem is of fundamental importance. Wait, sorry. Protein structure prediction can be used to determine the three-dimensional shape of a protein from its amino acid sequence. This problem is of a fundamental importance as the structure of a protein largely determines its function. Largely determines its function. Okay. However, protein structures can be difficult to determine experimentally. Considerable progress has recently been made by leveraging genetic information. It is possible to infer which amino acid residues are in contact by analyzing covariation in homologous sequences, which aids in the prediction of protein. We just read this. Cool. Proteins are at the core of most biological processes. As a function of a protein is dependent on its structure, understanding protein structures has been a grand challenge in biology for decades. Although several experimental structures determination techniques have been developed and improved in accuracy, they remain difficult and time-consuming. Fuck my wrist. As a result, decades of theoretical work has attempted to predict protein structures from amino acid sequences. This competition shit is a biennial blind protein structure prediction assessment run by the structure prediction community to benchmark progress in accuracy. In 2018, AlphaFold joined 97 groups from around the world and entering CASP 13. Each group submitted up to 5 structure predictions for each of 84 protein sequences for which experimentally determined structures were sequestered. Okay. Assessors divided the proteins into 104 domains for scoring and classified each as being amenable to template-based modeling. TBM, in which a protein with a similar sequence has a known structure, and that homologous structure is modified in accordance with the sequence differences, or requiring free modeling in cases in which no homologous structure is available. Okay, free modeling. That makes sense. It's like a piss in the wind, isn't it? With an intermediate FM-TBM category. Figure 1a shows that AlphaFold predicts more FM domains with high accuracy than in any other system, so the piss in the winds are quite accurate. Pretty good. I'm not very accurate myself, particularly in the 0.6 to 0.7 TM score range. The TM score ranging between 0 and 1 measures the degree of match of the overall backbone shape of a proposed structure to a native structure. The assessors ranked the 98 participating groups by the summed capped z-scores of the structures, z-scores, fucking hell, separated according to category. AlphaFold achieved the summed z-score of 52.8 in the FM category. Okay. But this is not CASP 14, so we're good. Okay. Compared with 36.6 for the next closest group. Combining FM and the TBM FM, TBM was, what was again? Template-based modeling. AlphaFold scored 68.3 compared with 48.2. AlphaFold is able to predict previously unknown folds to high accuracy. 1B. Can you guys see this? You can see this. You can see this. Cool. So the TM score, actually a bit cold. Got my little heater on. D3. TM score. AlphaFold. Got this one. That's interesting though. So does that note that the accuracy is not always better? What's the difference between these and this? Okay. Fine. That's fine. Just go with what we got. Despite using only FM techniques and not using templates, AlphaFold also scored well in the TBM category according to the assessors for Mueller zero-capped z-score, ranking 4th for the top 1 model or 1st for the best of 5 models. Much of the accuracy of AlphaFold is due to the accuracy of the distance predictions, which is evident from the high precision of the corresponding contact predictions. 1C. So the precision of the what's L? Can I have more information, please? Eh? Where does it say it? Much of the accuracy of alpha distance predictions is evident from the high precision L1, L2, L1, L2, L5. Number of contacts. Oh, where the amino acid contacts the other one. Anyway, so the precision for AlphaFold. Yeah, that looks good. Template-based ones are good, aren't they? So this is the problem. I need this one. Free modeling. It is amazing that we got such precision. I don't know. I guess that's the study of 5. Yeah, five contact points. I don't know what the L is. Okay, anyway. What happened to Figure 1? Because I don't remember looking at that. What does it talk about? Figure 1, the performance of AlphaFold in the CAS13 assessment. Okay. And extended data Fig 2a. Where's 2a? Sequence and MSA features. Tiled L by 1. 1D sequence and profile features. One-dimensional sequence and profile features. Okay. Goes through the Duke neural network. 220 residual convolution blocks. Convolution. Define convolution. A thing that is complex and difficult to follow. Residual difficult to follow blocks. Okay. Oh, that's annoying. I can see myself here. And then distance and torsion distribution predictions. Torsion. So distance and the rotation or folding distribution predictions. Out of all the possibilities it focuses in, I guess this is what this is trying to denote. The folding process illustrates. Okay, anyway, let's go back. So 2a. Right, figure 1. The performance of AlphaFold in the CASP13 assessment. Number of FM plus FM domains predicted for a given TM score threshold for AlphaFold and the other 97 groups. For the 6 new folds identified by the CASP13 assessors, the TM score of AlphaFolds compared with the other groups, together with the native structures. The structures of this is not available for publication. That's a shame. Is this going to be the same for everything? Alignment quality strip chart as function of position in sequence. Correctly aligned residues according to EQ. Residues aligned with -4, +4, window force shift according to EQv4p. EQv4p, this one. Residue aligned within -4 to 4, window force shift according to this. Residues aligned. Okay, so I see, but this is the output of it, isn't it? CASP13, 13th community-wide experiment on the critical assessment of techniques. This is the output. Okay, alignment quality strip chart as a feature. So who is this? So this is for this. Version 4. First models, all models. Where's AlphaFold? Mistyped. Oh, really mistyped. Where are we? Alpha. Okay, so that's not what this means. What is this meaning? Show groups. You have to select at least one group. Is it DeepMind? Oh, I don't know what group they are. I don't know. Okay, well, whatever. Precision for long-range contact prediction in CASP13 for the most probable L, L2 or L5 contacts, where L is the length of the domain. Okay, the distance distributions used by AlphaFold in CASP13, thresholded to contact predictions, are compared with the submissions by the 2 best-ranked contact prediction methods in CASP13, 498 Raptor X contact, and so threshold are compared with the submissions by the but used by AlphaFold. So where is their name in there? Raptor X. Let's see that one. Raptor X is one. Can I just see that? Quality. Is this Raptor X now? I can't quite tell. Show groups. Okay, this is it. So they were saying the submissions by the 2 best ranked, the distance distributions used by AlphaFold and CASP13 threshold to contact predictions are compared with the submissions by the 2 best ranked contact. This is for figure 1. Excuse me, cough, sorry. Ah, the scores are gone. Maybe I zoomed in too much. Yeah. Can I zoom in now? Nope. There we go. Cool. So AlphaFold is greatly better. Score cutoff. The domain count. Okay, cool. And 32 triplet res and all. Let's have a look at triplet res. Oops, that one there. Oh, what are they called? Oh, there we go. Triplet res? Nope, okay, well that's me misunderstanding there. Maybe they don't publish their results. The most successful FM approaches thus far have relied on fragment assembly. In these approaches, a structure is created through a stochastic sampling process. I do know what this means. I have to remind myself though. Random probability distribution or pattern that may be analysed statistically but may not be predicted precisely. Okay. Such as simulated annealing. Oh, you fucker. Oh, God. Ay, ay, ay. Is a heat treatment process which alters the is a probabilistic technique for approximating the global optimum of a given function. Specifically, it is a meta-heuristic to approximate global optimization in a large search space for an optimization problem. Look at this dude image. Right, let's read that again. That's quite a good graph. Annealing is a probabilistic technique for probabilistic techniques, like almost like quantum stuff. Approximate the global optimum of a given function. Okay, because there's no definitive. That minimizes statistical potential that is derived from summary statistics extracted from structures in the protein data bank. Oh. Hang on. What's that? Protein data bank. Search. Basic search. Click up here. Let's see. Structural basis of oligomerization and maintenance of thylakoid membrane integrity. Lipid binding protein. Synechocystis organisms. Expression system. E. coli. Oh, that's beautiful. General Research Foundation. Data snapshot. Electron microscopy. Yeah, I really want to buy one of these dudes. They're expensive. Metric. Clash score. Percentile ranks. Value. 3D report. Ooh. I like 3D. Oh, no way. Oh my god, that's beautiful. Oh my god, that is so beautiful. Wow, look at this. Oh, and you can click on the bits. What's the difference between that? B, Orth, F. Oh, ASP, Met. ASP. VAL. What about this one over here? Met. Glue. These look very similar. Oh my goodness, this is beautiful. How about, where's the arrow there? Okay, so there's the arrow and there's the arrow. So look at this. This. Gly 156, Gly 156, Orth 157, or 157, B Orth F, B Orth F, ASM 12, ASM 11. So is this ASM 10? ASM 10, got you. I can see you. 9, where's 1? 8, 7, 6, 5, 4, 3, 2, 1. ASM 1. Model 173Y. Yeah, that's the difference there. Okay, great. Wow, that is amazing. Whoa, what a database. Beautiful. Sequence. Oh my god. They are so kind to put this. Okay. Protein. What is this? Protein data bank. Okay. Let's go back to this. Paper. this was the, what's this called again? I've forgotten this. Abstract, that's the one. Okay, right, let's continue. So in fragment assembly, a structure hypothesis is repeatedly modified. Structure hypothesis repeatedly modified, typically by changing the shape of a short section while retaining changes that lower the potential, ultimately leading to low potential structures. Simulated annealing requires many thousands of such moves and must be repeated many times to have good coverage of low potential structures. In recent years, the accuracy of structure predictions has improved through the use of evolutionary covariation data. That are found in sets of related sequences. Sequences that are similar to the target sequence are found by searching large datasets of protein sequences derived from DNA sequencing and aligned to the target sequence to generate a multiple sequence alignment. Correlated changes in the positions of two amino acid residues. Correlated changes in the positions of two amino acid residues across the sequences of the MSA can be used to infer which residues might be in contact. Correlated changes in the position of two amino acid residues across the sequences of the multiple sequence alignment can be used to infer which residues might be in contact. Contacts are typically defined to occur when the beta carbon atoms of two residues are within 8A, what is this? Various sounds in several. Danish, Swedish for A. Ooh. Okay. We'll go for the ooh. Go for the ooh. Within 8 ooh of one another. Several methods, including neural networks, have been used to predict the probability that a pair of residues is in contact based on features computed from MSAs. Contact predictions are incorporated in structure predictions by modifying the statistical potential to guide the folding process to structures that satisfy more of the predicted contacts. Contact predictions are incorporated in structure predictions by modifying the statistical potential to guide the folding process to structures that satisfy more of the predicted contacts. That makes sense. Other studies have used predictions of the distance between residues, particularly the distance geometry approaches. Neural network distance predictions without covariation features were used to make the evolutionary pairwise distance dependent statistical potential, which was used to rank structure hypothesis. In addition, the quark pipeline used a template-based distance profile restraint for TBM. That makes sense. In this study, we present a deep learning approach to protein structure prediction, the stages of which are illustrated in Figure 2a. Yeah, let me just highlight that. Can I highlight this? Oh, I really hate... Is this a PDF? Fucking hate PDFs. God. 707, right. 2a was it? Figure 2a. Oh, you can click on it. Okay. Yeah, cool. We show that it's possible to construct a learned protein-specific potential by training a neural network to make accurate predictions about the structure of the protein given its sequence, and to predict the structure itself accurately by minimizing potential by gradient descent. The neural network predictions include backbone torsion angles and pairwise distances between residues. Distance predictions provide more specific information about the structure than contact predictions and provide a richer training signal for the neural network. So it's saying that the distance predictions provide more specific information about the structure than contact predictions. So rather than knowing how they contact, it's the distance between them using their physics model and all that stuff. They can get a much better structure output. Provide a richer training signal for the neural network. By jointly predicting many distances, the network can propagate distance information that respect covariation, local structure, and residue identities of nearby residues. The predicted probability distributions can be combined to form a simple principled protein-specific potential. We show that with gradient descent, it is simple to find a set of torsion angles with gradient descent. It is simple to find a set of torsion angles that minimizes this protein-specific potential using only limited sampling. We also show that whole chains can be optimized simultaneously, avoiding the need to segment long proteins into hypothesized domains that are modeled independently, as is common practice. The central component of AlphaFold is a convolutional neural network that is trained on PDB structures to predict the distances dij between the C-beta atoms of pair ij of residues of a protein. The central component of AlphaFold is a convolutional neural network that is trained convolutional. Yeah. Convolutional. Refers to both the result function and to the process of computing it. The central component of AlphaFold is a convolutional neural network that is trained on PDB structures, protein... Wait, what was PDB again? Where are we? Protein data bank. PDB, okay. It's trained on the protein data bank. Network that is trained on protein data bank structure to predict the distances dij between the C-beta atoms of pairs, ij of residues of a protein. The central component, this is so important, this part, the central component of AlphaFold is a neural network that is trained on the structures in that database to predict the distances d, ij, which is what route it took, between the C-beta atoms, the distance between each neural network's output, atoms of pairs, C-beta atoms of pairs. So to predict the distance between the C-beta atoms of pairs, ij, each indice of residues of a protein, on the basis of a representation of the amino acid sequence, S, of a protein and features derived from the MSAs of that sequence. The network, which is similar in structure to those who use for image recognition tasks, predicts a discrete probability distribution, P, the probability of dij, so the distance between those two nodes, okay, wait, wait, let me just get that mathematical symbol because I can't remember what that means, and I have looked it up before, it's really annoying. Mathematical symbol, pipe. The vertical bar is a glyph with various uses in computing and typography. It has many blah blah blah. Mathematics, magnitude. This one, conditional probability. Okay, the probability of dij to S of a protein acid sequence. The probability of the distance. Distance given this acid sequence. And MSA's multiple sequence alignment. So the probability of the distance of the sequence and the multi-sequence alignment of S. For every ij pair in any 64 by 64 region of the L by L distance matrix, as shown in figure 2b. Why can I not select that? It's so annoying. I want to like mark it. 2b, 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 or not to be 2b. L by one dimension sequence of bar features. L by L2D covariation features. 64 by 64 matrix. Distance and torsion distribution predictions. Okay, that makes sense. The full set of distance distribution predictions constructed by combining such predictions that covers the entire distance map is termed a distogram. Or distance histogram. Example distogram predictions for one Cas protein. 20955 are shown in figure 3CD. The probability, a logarithmic scale, the distance of like that. Let me just try and figure this out. 10 minus 2, 10. What? Distance and probability of that distance. And it's saying the distogram predictions for one Cas protein are shown in the mode of the distribution. Can be seen to closely match the true distances. Excuse me, figure 3b. So D, distance, distance and of this sequence, is it? No. What am I looking at here? The probability and distance is equal to this or close to it. So let's take 10, 0. So this must be the picture that they take of the actual protein, the template. Okay, whatever. The full set of distance distribution predictions constructed by combining such predictions that covers the entire distance map is termed a distogram. Yep. The mode of the distribution 3C can be seen to closely match the true distances 3B. Example distributions for all distances to one residue 29, are shown in figure 3D. We found that the predictions of the distance correlate well with the true distance between residues figure 3D and 3E. 3D. 29 did it say? 29, yeah. 3D, 3E. Okay, 29. Where is it? There it is. 28. Wait, what? It says Okay. Example histogram predictions can be seen to closely match the true distances. Example distributions for all distances to one residue 29, are shown in figure 3D. 3D 29. The Moser distribution can be seen to closely match the true distances. Example, distributions for all distances to one residue are shown in figure 3D. No, they're not. We found that the predictions of... What? We found that the predictions of the distance correlate well with the true distance between residues figure 3e. Furthermore, the network also models the uncertainty in its predictions figure 3f. The mode of prediction, U, and true distance. So the true distance, denoted by which colour I'm not sure. I'm guessing orange. Distance 6. Yeah, it's got some grouping with this. I see. These are interesting though. This is the true distance here, look. I wonder why... Yeah, a little bit insider I think. I don't quite get that, but... So, when the S.D of the predicted distribution is low, the predictions are more accurate. Okay, this is also evident in figure 3D, in which more confident predictions of the distance distribution, higher peak and lower SD of the distribution, tend to be more accurate, the true distance close to the peak. Broader, less confidently predicted distributions still assign probability to the correct value, even when it is not close to the peak. The high accuracy of the distance predictions, and consequently the contact predictions, figure 1c, come from a combination of factors in the design of the neural network and its training data augmentation. Feature representation, auxiliary losses, cropping, and data curation, see methods. To generate structures that conform to the distance predictions, we constructed a smooth potential v distance by fitting a spline to the negative log probabilities, and summing across all of the residue pairs. To generate structures that conform to the distance predictions, we constructed a smooth potential v distance by fitting a spline to the negative log probabilities and summing across all of the residue pairs. So they're saying that they've done this. Oh, let me just grab this. I probably need to grab that a bit better like this. I need like a whiteboard or something. This feels like, cause it's kind of giveaway. It's like I just want to be able to like draw quickly, but then I need to set up a camera so you can see it. Okay, gotcha. So it's saying, yeah, to generate structures that conform to this position, we constructed a smooth potential v distance. So let's just draw that. So you got by constructing a smooth potential v distance by fitting a spline to the negative log probabilities and summing across all of the residue pairs. Wait, so it doesn't denote it here. Just move that over here for a second. C methods. We parameterize protein instruction. So they've got the sum of all the residue pairs. So let's say this is a pair. Well, actually, a bit more like that other actual data, but like this. And they've got like these kind of squiffy things here. And they've got fit the spline to negative log probabilities. Log, let me just get that, mathematics, log, log, okay. In mathematics, the logarithm is the inverse function to exponentiation. That means the logarithm of a given number x is the exponent to which another fixed number, the base b, must be raised to produce that number. So the inverse of exponent. That means the logarithm of a given number x is the exponent to which the inverse of an exponential. So let's say if it goes up here exponentially, what is the inverse to get that back down? What is mathematics log used for? It means by logarithm of a given number is the exponent to which another fixed number, the base b, must be raised to produce that number x. In the simplest case, the logarithm counts the number of occurrences of the same factor. In the simplest case, the logarithm counts the number of occurrences of the same factor in repeated multiplication. Since 1000 equals 10 by 10 by 10 by 10, the logarithm base 10 of 1000 is 3. Or log 10, 1000. 1000 equals 10 by 10 by 10 is equal 10 to the power of 3. So the logarithm base of 10 is 3, or log 10, 1000 equals 3. The logarithm of x to base b is denoted as log b of x, or without parentheses, log bx, or even without the explicit base, log x, when no confusion is possible, or when the base does not matter, such as a big O notation. More generally, explanation allows any positive real number as base to be raised to any real power, always producing a positive result. So log b of x for any two positive real numbers b and x, where b is not equal to 1. For any two positive real numbers b and x where b is not equal to 1, is always a unique real number. I'm reading off the screen, sorry. Okay. Fine. I don't know if I can draw this, I don't quite understand it. Like its purpose. What's the intuition for a log? It's like, people are like, this is what it does, but what's the use for it, you know? In solving exponential equations, some examples of this include decibel measures, earthquakes, Richter scale, the brightness of stars, and chemistry. Here we go. Let me share. I've got the wrong thing, but yeah, let me try and share this. Yeah, I've seen many different kinds of functions. You know that each one could be used to model some kind of situation in the real world. Exponential and logarithmic functions are no exception. Much of the power of logarithms is their usefulness in solving exponential equations. Some examples of this include sound, decibel measures, earthquakes, Richter scale, the brightness of stars, and chemistry, pH balance, a measure of acidity and alkalinity. Let's look at the Richter scale, a logarithmic function that is used to measure the magnitude of earthquakes. The magnitude of an earthquake is related to how much energy is released by the quake. Instruments called seismic graphs detect movement in the earth. The smallest movement that can be detected shows on a seismograph as a wave with amplitude A0. A, the measure of the amplitude of the earthquake wave. A sub 0, the amplitude of the smallest detectable wave or standard wave. From this you can find R, the Richter scale measure of the magnitude of the earthquake using the formula. From this you can find the Richter scale measure of the magnitude of the earthquake using the formula. R is the log of A over A0. The intensity of an earthquake will typically measure between 2 and 10 on the Richter scale. Any earthquakes registering below a 5 are fairly minor. They may shake the ground a bit, but are seldom strong enough to cause much damage. Earthquakes with Richter scale rating of between 5 and 7.9 are much more severe, and any quake above an 8 is likely to cause massive damage. The highest rating ever recorded for an earthquake is 9.5 during the 1960s. Example, an earthquake is measured with a wave amplitude 392 times as great as A sub naught. What is the magnitude of this earthquake using the Richter scale to the nearest tenth? An earthquake is measured with a wave amplitude of 392 times as great as A sub naught. What is the magnitude? It's going to be 392 over A sub naught. 392 times A sub naught divided by A sub naught and a log of that. Yeah, but that doesn't really say much. Like, just have this number then. A difference of 1 point on the Richter scale equates to a tenfold difference in the amplitude of the earthquake. Yeah, but once again, this is just like some unit output, right? Look at the example just shown. In that example, the wave amplitude of the earthquake was 292 times normal. What if it were 10 times that, or 3,920 times normal? To find a measurement of that size earthquake on the Richter scale, you find log 3920. Calculate gives a value of 3.5 or 3.6. Around blah, blah, blah. Sound is measured in a logarithmic scale using a unit called a decibel. The formula looks similar to the Richter scale. 10 log of P over P0, where P is the power of intensity of the sound and the P0 is the weakest sound that the human ear can hear. One hot water pump has a no— yeah, but why? This is crap. Does not tell me why that's important. Future work with logarithm. Why do we use log? Oh, here we go. Someone knows what they're doing. Let's see. Go away. There are two main reasons to use logarithmic scales in charts and graphs. The first is to respond to skewness towards large values, i.e. cases in which one or a few points are much larger than the bulk of the data. Skew. The second is to show percentage change or multiplicative factors. First I'll review what we mean by logarithms, then I will provide more detail about each of these reasons to give examples. To refresh your memory of school math, logs are just another way of writing exponential equations, one that allows you to separate the exponent on one side of the equation. The equation 2 to the 4 equals 16 can be written as log 2 16 equals 4. Okay, I see. Okay, I see. I understand now. Sorry, thanks for going along with me then. So this is saying v distance by fitting a spline to the negative log probabilities and summing across all of the residue pairs. Okay. Negative log probabilities and summing, because they want to find what the, wherever that thing was now has gone. Is that one of my Chrome tabs? Yeah. They want to know what that exponent was. Okay, I see. Summing across all of the residue pairs. Negative log. Okay, fine. So this is let's see. So we parameterize protein structures by the backbone torsion angles, phi and whatever that is, of all residues and build a differentiable model of we parameterize protein instructions by the backbone torsion angles of all residues and build a differentiable model of protein geometry. X equals G as a function of phi and that to compute the C beta coordinates xi for all residues I, and thus the interresidue distances dij. So this is distances of each layer. No, each layer. No, each dimension of that layer is equal to the magnitude of xi minus xj to compute the coordinates xi for all residues I. This is interesting. For each structure, and express v distance as a function of v and... What is this again? Is that gamma? Psi and phi. Psi and phi. Let's see. I think that's phi. Psi and phi. Phi and c. C? Psi. Psi? Phi? Can't remember. Sci-fi? C. Psi. Sci-fi. For a protein with L residues, this potential accumulates L2 terms. So for a protein with L distance residues, this potential accumulates L to the power of 2 terms from marginal distribution predictions. To correct for the overrepresentation of the prior, we subtract a reference distribution from the distance potential in the log domain. We subtract a reference distribution from the distance potential in the log domain. Okay. We subtract the reference from the potential. The reference distribution models the distance distributions. Probability of dij of length. Probability of the distance of those nodes of the length. The probability of those distance nodes of the length. Independent of the protein sequence and is computed by training a small version of the distance prediction neural network on the same structures, without sequence or MSA input features. A separate output head of the contact prediction network is trained to predict discrete probability distributions of backbone torsion angles. Oh, yeah. So probability of phi of I and psi of I of the sequence. What was that again? S was amino acid sequence. The probability of that sequence and the multi-sequence amino something. No, it wasn't. It was the. It says up here multiple sequence alignment. The probability of phi and psi of the sequence and then the multiple sequence alignment. Okay, I don't quite get that. Probability of that. Probability of that. So, where... But we'll come back to this. Okay, let me just finish off this part. So after fitting a von Mises distribution, right, here we go. Distribution. Ludwig Heinrich Edler von Mises, German, 1881-97, was an Austrian school economist, historian, logician, and sociologist. Mises wrote and lectured extensively on the societal contributions of classical liberalism. He's best known for his work on praxeology, study of human choice and action. Mises immigrated from Austria. Many of its alumni, including Friedrich, immigrated from. Mises' private seminar was a leading group of economists. Many of its alumni, including Friedrich Hayek and Oskar Morgenstern, immigrated from Austria to the United States and Great Britain. Mises has been described as having an early life in Europe, work in the States. Okay. So wrote and lectured extensively on the. So what is this? Von Mises distribution. Alright. Let's look at that. In probability theory and directional statistics, the von Mises distribution, also known as the circular normal distribution or Tikhonov distribution, is a continuous probability distribution on the circle. It is close approximation to the wrapped normal distribution, which is the circular analogue of the normal distribution. A freely diffusing angle theta on a circle is a wrapped normally distributed random variable with an unwrapped variance that grows linearly over time. Okay, I see. A freely diffusing angle theta on a circle is wrapped normally distributed random variable. Is a wrapped normally distributed random variable with an unwrapped variance that grows linearly over time. On the other hand, the von Mises distribution is the stationary distribution of a drift and diffusion process on a circle in a harmonic potential with the preferred orientation. The von Mises distribution is the maximum entropy distribution for circular data when the real and imaginary parts of the first circular moment are specified. The von Mises distribution is a special case of the von Mises-Fisher distribution. So this is saying probability density function minus pi over 2, 0, pi over 2, and probability density function. Fine, I don't understand that. I don't understand that at all. Let me go back to my article. This is used to add a smooth torsion modeling term. I mean, I can get some intuition from it, I suppose. Because as it gets closer to the number, it's kind of peaked out. Pi over 2 distribution. Finally, to prevent steric clashes, we add the v-score2 smooth score of Rosetta to the potential, as this incorporates a van der Waals term, van der Waals. We used multiplicative weights for each of the three terms in the potential. However, no combination of weights noticeably outperformed equal weighting. Van der Waals. Van der Waals forces include attraction and repulsion between atoms, molecules and surfaces, as well as other intermolecular forces. They differ from covalent and ionic bonding in that they are caused by correlations in the fluctuation polarizations of nearby particles. That's great. Forces include attraction and repulsion between atoms, molecules, as well as they differ from covalent and ionic bonds in that they are caused by correlations in the fluctuations. So the force is equal to the negative of the Haymaker constant. Spherical bodies radii. So it's the constant multiplied by two radiuses multiplied together in ratio of the first radius plus the second radius times 6 distance between the surfaces squared. That's beautiful. Okay. We use multiplicative weights for each of the three terms in the potential. However, no combination of weights noticeably outperformed equal weighting. As of all the terms in the combined potential, V total phi psi, V of psi as all of the terms in the combined potential are differentiable functions, it can be optimized with respect to these variables by gradient descent. Here we use this. Here we use this. Structures are initialized by sampling torsion values from this. Figure 2c illustrates a single gradient descent trajectory that minimizes the potential, showing how this greedy optimization process leads to increasing accuracy and large-scale conformation changes. Greedy optimization process. Greedy, is that meaning that it's more parameters or less? Optimization. Its greedy optimization process leads to increasing accuracy and large-scale conformation changes. The secondary structure is partly set by the initialization from the predicted torsion angle distributions. The overall accuracy improves quickly, and after a few hundred steps of gradient descent, the accuracy of the structure has converged to a local optimum of the potential. This is interesting. This is their weighting. Or it's their sigmoid. $7.08. Okay, let's just take a screenshot of that. Let's put that here. I'm gonna have to stop now because I need some food. We got here. Right, that's enough for today. How long have I been on it? Oh my god, so long. Okay, yeah, time to go. Thanks for watching if you were. If you weren't, thanks for not watching, I guess. And keep loving, keep learning. Bye for now.